Good evening. Let's explain the research on interplasmid transfer of antibiotic resistance genes accelerates antibiotic resistance in bacterial pathogens. What is antibiotic resistance? Antibiotic resistance is when bacteria evolve, becoming able to defeat the drugs designed to kill them. This happens because some germs naturally survive the treatment and multiply, creating a resistant population. These resistant infections are then much harder or impossible to treat with common medicines. The overuse and misuse of antibiotics significantly accelerates this natural evolutionary process globally. Responsible for transfer. A plasmid is a small, typically circular, double-stranded DNA molecule that is physically separate from a cell's chromosomal DNA and can replicate independently. Plasmids are primarily found in bacteria and archaea, but also sometimes in eukaryotes. They carry a small number of non-essential genes that often confer beneficial traits to the host cell, such as antibiotic resistance. Think of bacterial chromosome as the main hard drive, containing the essential operating system for survival. A plasmid is like removable USB drive. It contains extra, non-essential genes that give the bacteria advantages, especially under stressful conditions. Mechanism used in research. Cell can host multiple plasmids. P1, the donor, carries the resistance gene, ARG, and a jumping gene, called IS26. IS26 pastes the ARG into the recipient plasmid. Now, the resistance gene is duplicated across two different plasmid types. To prove this internal swapping, the scientists used two powerful methods: massive computer analysis and a custom lab experiment. The GFP marker proof. They engineered a plasmid carrying the IS26 element linked to a gene for gentamicin resistance, and attached a green fluorescent protein, GFP, marker. The successful jump. To show the IS26 GFP unit jumping to a second plasmid, P2, they rigged the system so that the bacteria would only glow green if the IS26 successfully cut the package out and pasted it onto a second, clean plasmid. Mechanism verified. When the cells turned bright green, it confirmed that the jumping mechanism driven by IS26 was rapid, real. The impact. P2, now a superplasmid, is shown leaving the cell via a tube conjugation. The plasmid is now a superplasmid, armed with multiple resistance genes. It can quickly spread this resistance to other bacteria. The findings of research and results. 86.6% of ARGs showed signs of recent transfer. Significance. This is the study's most critical finding. It means that the movement of antibiotic resistance genes, ARGs, is not rare or accidental. It is a normal, dominant mechanism. The vast majority of resistance genes are constantly jumping, suggesting that plasmids are always mixing their genetic contents rather than just staying fixed. Second finding. Transfer is much more frequent in clinical plasmids. Significance. This links the molecular mechanism to the public health crisis. Clinical plasmids, those found in hospitals and human infections, swap genes more frequently than environmental plasmids, from soil or water. This is driven by the intense selection pressure of heavy antibiotic use in clinical settings, forcing the bacteria to rapidly assemble resistance genes to survive. Third finding. Antibiotic resistance genes jumping overwhelmingly favors compatible plasmids. Significance. This confirms the mechanism occurs internally. Compatible plasmids are types that can coexist peacefully within the same single bacterial cell. The high rate of transfer between them suggests that the gene swapping happens while the two plasmids are in close proximity, rather than relying on the plasmids moving between different cells. Fourth finding. IS26 is the main driver, 63.1% of events. Significance. The study identified the key molecular tool. The IS26 element, an insertion sequence, or mobile genetic element, is that drives most of the gene jumping. Identifying this specific element makes it a clear target for future strategies designed to block resistance spread. The results. Stopping the IS26 jumping mechanism is the new goal. This is the practical conclusion and call to action for the scientific community. Since the IS26 element is the main tool driving the assembly of superplasmids, New drugs or genetic therapies should focus on deactivating or blocking the function of IS26, stopping the resistance genes from jumping and multiplying.